Imagine you're walking through a really dense, dark forest in the dead of night.
Oh, yeah, like pitch black.
Exactly. The canopy above you is so thick it completely blocks out the moon, and your eyes are just straining to adjust.
Right.
And then, just as you're about to give up and turn back, you see something that just seems impossible.
The foxfire.
Yeah. Right at the base of this old, decaying oak stump, there's a piece of wood glowing with this eerie, steady bluish-green light.
It's such a wild visual.
It really is. And it's not a hidden battery pack. It's not like some discarded LED from a flashlight. What you're looking at is entirely biological.
A living, breathing, wood-eating organism.
Exactly. So, welcome to today's deep dive. We are exploring the hidden world of this ancient and honestly mind-bending fungus called Desarmillaria tabescens.
Or most people probably know it as the ringless honey mushroom.
Right, the ringless honey mushroom.
Yeah.
And to really understand what's happening inside that glowing wood, we spent the last few weeks digging through a massive mountain of source material.
Oh, yeah. A lot of reading for this one. Seriously. We're talking dense phylogenetic research, molecular clocks, pathology reports on forest devastation.
The genomic sequencing data alone was wild.
Yeah, it reads like a pharmaceutical catalog. Plus, there's these incredibly futuristic materials science studies out of Switzerland.
So we're synthesizing all of that today to shortcut your path to understanding this thing.
Because it's not just a neat glowing mushroom you might stumble across in your backyard.
No, not at all. By understanding this specific fungus, you're actually looking at the deep history of Earth's climate.
Right?
You're looking at, literal chemical warfare happening beneath the forest floor.
And maybe the craziest part, the foundational science for the future of sustainable zero-energy lighting.
Yeah, living architecture. It's wild.
So before we get to the futuristic stuff, like human scientists hijacking this fungus to build biological light bulbs, we have to understand what it actually is.
We have to rewind the clock way back.
Yeah, because we're looking at a creature that is basically a relic of a lost world.
We need to go back roughly 51 million years here to the Paleocene-Eocene thermal maximum, right? The PTM.
Exactly. So to put that in perspective for you, the dinosaurs are long gone, but the Earth we know today doesn't exist yet.
Right, it was way hotter.
Yeah, the PTM was a period of extreme, rapid global warming. We're talking massive, dense tropical rainforests stretching almost to the poles.
Like palm trees in Alaska.
Palm trees in Alaska, yeah. It was wet, it was humid, it was a global sauna. And it was in this sweltering hothouse environment, likely somewhere in Eurasia, that the armillarioid lineage first evolved.
Which is the family tree our fungus belongs to.
Right.
And the research points out something that feels almost poetic. Desarmillaria tabescens still carries the genetic memory of that ancient greenhouse world.
Yeah, it's considered a thermophilic species.
Thermophilic meaning heat-loving.
Heat-loving, yeah. Even today, millions of years later, when you put it in a lab incubator, it thrives at temperatures that would just stress out other temperate fungi.
The sources said its optimum growth is around 28 to 30 degrees Celsius.
Exactly, and that thermal preference isn't a new adaptation, it's an evolutionary hangover.
It's built for a sauna because it evolved in one.
Right, but obviously the Earth didn't stay a sauna forever.
No, the climate eventually shifted.
Drastically. Moving out of the Eocene and into the Oligocene epoch, the global climate cooled down. Ice caps formed, and vast regions became way more arid.
And if you're an organism built for tropical humidity,
it's an existential crisis.
Right, your whole world is disappearing.
So in response to this drier, harsher climate, a newer branch of this fungal family tree evolved to dominate the landscape.
The Armillaria species.
Yeah, the true Armillaria. They developed specialized physical adaptations to survive the cold and dry summers. Meanwhile, our original lineage, the Desarmillaria,
it was forced to retreat.
Yeah, it couldn't compete everywhere anymore. It became restricted to highly specialized ecological niches.
Like deep, waterlogged root systems or humid ravines. Places where its warm, wet preferences were still an advantage.
Exactly. It's a survivor hiding in plain sight.
And looking at the phylogenetic data from our sources, its family tree has some seriously weird branching.
Oh, absolutely.
There's this concept discussed in the evolutionary papers called the gastromycetation trajectory.
Such a great term.
Yeah, so our ringless honey mushroom is an early diverging lineage, and the genome shows it's a sister species to something completely bizarre.
The genus Guyanogaster.
Right, Guyanogaster.
To appreciate how strange this is, you have to look at normal fungal reproduction. Most classic mushrooms are what we call ballistosporic.
Meaning they shoot their spores?
Yeah, they have gills underneath their caps, and they actively shoot spores into the open air using a microscopic catapult system powered by a single drop of water.
Which is incredible on its own.
It is. It's biological engineering designed to catch the wind.
And gastromycetation is when they lose that ability.
Exactly. It's a fascinating one-way street in fungal evolution. Sometimes, due to environmental pressures, a lineage will slowly evolve away from that open gill structure and enclose itself completely.
Like a puffball or a truffle.
Right. It becomes a closed underground mass. It loses the ability to forcefully discharge spores, so it relies on animals digging it up and eating it to spread the seeds.
And once it goes down that path, it never goes backward.
Never. You never see a truffle evolve back into a gilled mushroom.
So, millions of years ago, the ancestor of our glowing fungus hit a fork in the road.
One branch stayed above ground, kept its gills, and gave us Desarmillaria.
And the other branch.
Underwent radical gastromycetation. It completely changed its body plan and became Guyanogaster, which is this highly specialized truffle-like fungus only found attached to specific tree roots in South America.
Wow. And genetic sequencing proves these two wildly different organisms share a recent common ancestor.
It's a testament to how extreme evolutionary divergence can be.
Wait, let's pause there, because I had a major point of confusion looking at this evolutionary timeline.
Okay, what is it?
So the common name for our fungus is the ringless honey mushroom. Now, those newer, more dominant cousins, the Armillaria species, they all have rings on their stems.
Right.
And that ring is the remnant of an annulus, a partial veil of tissue that protects the delicate young gills as the mushroom develops, right?
Correct. It acts as a protective shield.
Okay, so if the climate was getting colder and drier and harsher, dropping your protective armor seems like the terrible evolutionary move.
Yeah, it does.
Why on earth would Desarmillaria ditch the ring and become ringless when the world was getting more dangerous?
Well, it's easy to look at it that way, but that assumes it had a ring and lost it.
Oh.
The reality revealed by the genetic mapping is the other way around. Desarmillaria didn't lose its ring. It never had one.
Wait, really? It never had one.
Never. The lack of an annulus is actually the ancestral trait for this whole section of the family tree.
Okay, so it's the baseline model.
Precisely. Remember, it evolved during the PTM when the environment was incredibly wet and warm. It didn't need a protective veil over its gills because the ambient air was already saturated with moisture.
Ah, so there was no risk of drying out in the wind.
Exactly. The protective veil, that ring, evolved later in the Armillaria lineage as a brilliant adaptation to the drier, colder climates that followed.
Oh, that makes so much sense.
Yeah, the newer mushrooms needed that covering to survive the new, harsh world. Desarmillaria just stayed true to its ancient ringless form.
Which perfectly sets up this massive modern-day taxonomic drama that the research papers dig into.
Oh, the family reunion drama.
Yeah, because for decades, scientists were looking at these ringless honey mushrooms growing on dead trees in Europe, across Asia, and in North America, and they just assumed, yep, that's all one big happy global species.
Right, Desarmillaria tabescens worldwide.
But the recent molecular data completely shattered that assumption.
It's one of the great cases of mistaken identity in modern mycology. It was driven by what we call vicariance.
Like continental drift separating populations.
Exactly. Vicariance is the geographical separation of a single biological population by a massive physical barrier, like a new ocean or tectonic shift, which eventually results in closely related but distinct species.
Because they can't interbreed anymore.
Right. For years, relying purely on the outward shape of the mushroom, everyone was convinced they were looking at a single cosmopolitan fungus.
Until they applied modern multi-locus phylogenetic analyses.
Yeah, the paradigm shifted overnight.
Multi-locus, meaning they aren't just looking at one quick genetic marker, right? They're diving deep into multiple highly stable sections of its DNA.
They examined 5 independent, highly conserved housekeeping genes. These control fundamental cellular processes, so they mutate at a very slow, predictable rate. It acts like a highly accurate molecular clock.
And they looked at portions of the 28S ribosomal RNA, the translation elongation factor, 1-alpha actin.
Yeah, and by analyzing the tiny accumulative mutations across those 5 separate genetic loci, they built an incredibly robust family tree.
And what did that molecular clock actually reveal about these mushrooms?
It proved definitively that the Eurasian populations and the North American populations are completely reproductively isolated.
So they might look identical from a few feet away, but genetically totally distinct.
Totally. In the phylogenetic analysis, the separation in certain genes showed 100% bootstrap support.
Of 100% bootstrap support. I love that detail. That's the statistical equivalent of a paternity test coming back with zero margin of error.
Exactly. It means the computer ran the evolutionary simulation thousands of times, and every single time it concluded these are separate species.
So they were separated by oceans, and over millions of years, their DNA drifted apart.
Yep. So the true Desarmillaria tabescens is now officially restricted to Eurasia.
And the North American cousin had to be officially renamed.
Right. It's now scientifically known as Desarmillaria caespitosa.
And the fascinating part is, once the geneticists told the morphologists that they were looking at two different species, they went back and looked closer.
Put them under high-powered microscope.
Yeah, and suddenly they started noticing all these physical differences they had overlooked for a century. They aren't actually identical twins.
Not at the microscopic level, no. The true Eurasian tabescens has narrower basidiospores and primarily regular cylindrical chylocystidia.
Which are those sterile cells that line the edge of the gills to keep them separated, right?
Right. But the North American caespitosa has significantly wider spores on average, and its chylocystidia are much narrower and often irregular, mixed, or even coralloid.
Coralloid meaning branched like microscopic coral reefs.
Exactly.
Okay, so we have this ancient tropical survivor, biologically isolated across continents, hiding out in specialized wet niches and holding on to its ringless form for 50 million years.
That's the baseline, yeah.
But this history didn't just shape the size of its spores. It equipped this fungus with a highly specialized toolkit to dominate modern forests.
Oh, absolutely. It's a powerhouse.
Which transitions us to the ecological reality of this organism. Because while it might be a relic, it is not helpless. It's a dominant force on the forest floor.
Dominant is the precise word, because it's categorized as a facultative necrotroph.
Meaning it's flexible, it can be a peaceful recycler, or it could be a killer.
Total ecological versatility. It can live a perfectly quiet life as a saprobe, just breaking down the dead, rotting wood of a fallen oak tree, cycling nutrients.
Just minding its own business.
Right. But if the opportunity arises, it can flip a biological switch and become a primary pathogen, actively attacking and killing healthy trees.
It's a major cause of armillaria root rot. And the sources say it triggers a terrifying defensive response in trees called gummosis.
Yeah, you see this heavily in oak trees and introduce species like eucalyptus.
The pathology reports on this are intense. The tree realizes its root system is being invaded, and in this desperate panic, it starts bleeding thick, sticky sap from its trunk.
Literally trying to flush the infection out of its vascular system.
It's a visible sign of a brutal subterranean war. But the most interesting aspect of how our fungus fights this war is what it lacks.
Right, the missing infrastructure.
Because when you read about the major honey mushrooms, they're famous for building these massive underground networks of black shoestring rhizomorphs.
The foraging rhizomorphs, yeah.
Right, these tough, root-like structures that physically forage through empty soil for yards, hunting blindly for new trees to infect.
It's how the famous humongous fungus in Oregon covers square miles of forest.
Exactly. But the sources explicitly state that our fungus rarely, if ever, produces those foraging rhizomorphs in nature.
It just doesn't build that underground infrastructure to cross open soil.
But logically, that doesn't make sense to me. If it doesn't build those structures, how does it infect new trees?
Right, if it can't sprint.
Yeah, if it can't sprint across the forest floor, how is it such a widespread pathogen?
Because it doesn't need to sprint if it knows how to hold on.
Oh, I like that.
Instead of expending massive cellular energy, sending exploratory rhizomorphs through dirt, it relies almost entirely on a physical process called root grafting.
Root grafting, okay.
Beneath the soil, the root systems of trees are constantly growing, shifting, overlapping. Eventually, the infected root system of a dying tree makes direct physical contact with a healthy root system.
And as the roots rub together,
The bark wears thin, the cambium layers touch, and they physically fuse.
and the fungus is just waiting at the point of contact.
Exactly. The mycelium waits for that bridge to form, and then it simply crosses over. It requires direct, prolonged physical contact.
It's like a slow-motion zombie bite. It can't run after you, but if you brush up against it in the dark, the infection transfers.
Perfect analogy. And once that physical bridge is crossed, the fungus unleashes its genomic weaponry.
Which brings us to its DNA.
Yeah, the genome of Desarmillaria tabescens is about 50 megabases in size, which is noticeably smaller than other Armillaria species, which can be 70 megabases or larger.
So it's compact.
Smaller but lethally optimized. It possesses a dense arsenal of genes coding for plant cell wall-degrading enzymes, primarily pectinases and lignases.
Those are the molecular sledgehammers, right?
Precisely. To eat a tree, you have to break its structural integrity. Pectinases degrade pectin, the biological glue holding plant cells together.
And lignases break down lignin, which gives wood its rigidity.
The fungus secretes a chemical cocktail that digests the tree's physical defenses from the inside out, turning solid wood into a nutrient-rich slurry.
But the truly staggering part isn't just the destructive power, it's where the fungus is capable of wielding that power.
Ah, the waterlogged wood?
Yes. The ecological data highlights a survival mechanism that is just brilliant. It often infects wood that is highly saturated with water.
Flooded roots, drainage ditches.
Right. Environments where the oxygen levels are incredibly low. For most fungi, that much water is a death sentence. They'd just drown.
But our fungus survives because it builds a snorkel.
A microscopic biological snorkel. I couldn't believe this when I read it.
It literally builds a snorkel. While it doesn't build long, foraging rhizomorphs in the soil, it does build highly specialized rhizomorphs internally within the wet wood.
Okay.
And when the wood is heavily saturated, the fungus forms a thin, dense, protective rind of melanin on the surface, right where it meets the air.
Melanin. As in the exact same pigment found in human skin.
The very same. Now, in humans, it protects our DNA from UV radiation. In fungi, it's highly hydrophobic, meaning it actively repels water.
So it acts as a waterproof shield.
Exactly. It prevents the external water from flooding the internal fungal network. Plus, it has antimicrobial properties, creating a sterile barrier against competing bacteria.
Okay, so it seals itself inside this dark, waterproof shield. But it still needs to breathe.
And that's where the snorkel comes in. The rhizomorphs, living deep inside the suffocating wood, connect to the surface by growing thin, specialized air pores.
And they punch right through that melanin rind.
Right, in tiny little tufts. Because the melanin keeps the water out, these pores remain dry, acting as open gas exchange channels.
They're actively drawing atmospheric oxygen from the open air down into the saturated wood.
Breathing underwater, it allows it to colonize low-oxygen niches where every other competing fungus just suffocates and dies.
It turns the worst real estate in the forest into its own private fortress.
It's an evolutionary masterpiece.
It really is. But here's where the narrative loops back to that eerie image we started with.
The foxfire.
Yes. That exact process, aggressively eating waterlogged wood and pulling oxygen down into the dark, is the biological trigger for its most magical trait, bioluminescence.
A phenomenon that has captivated humans for millennia. Aristotle and Pliny the Elder wrote about it.
Fairy fire,
Yeah.
cold fire. But we need to clarify a widely unknown fact here. If you go out to the woods and find a beautiful cluster of ringless honey mushrooms and take them into a pitch-black room,
You're going to be very disappointed.
right, the mushrooms themselves do not glow.
Not a single photon. The fruiting body, the cap, stem, gills, completely dark. The light emission is entirely restricted to the vegetative mycelium hidden inside the wood.
So to see the light, you have to break open a rotting log in the dark.
Exactly.
So what's actually happening inside that wood to create this light? The researchers call it the caffeic acid cycle.
It's a beautiful chemical cascade. Well, it starts with the fungus acting as a white rot decomposer. As it releases these ligninases to break down the tough lignin in the wood, it leaves behind chemical rubble.
Right, the phenylpropanoids.
Exactly. And it doesn't waste that rubble. It absorbs those phenylpropanoids and metabolizes them into an intermediate molecule called hispidin.
Okay, phenylpropanoids to hispidin.
And it uses another enzyme to convert that hispidin into the star of the show, luciferin.
Luciferin, the magic ingredient.
Yeah, the chemical potential waiting to be released. In the presence of an enzyme called luciferase, and crucially, in the presence of oxygen, the luciferin is rapidly oxidized.
And that oxidation is an exothermic reaction. It releases energy.
But not as heat. It releases it as a single photon of visible light, at a wavelength of roughly 560 nanometers, which we perceive as that steady bluish-green glow.
This connects the biology so perfectly, because that snorkel system we just talked about,
Yeah.
the one pulling oxygen into the wet wood, that oxygen is the final spark to ignite the luciferin.
Without the melanin rind and the pores, there's no oxygen. No oxygen, no oxidation, no glow.
But this raises a massive evolutionary question. The sources touch on this, and it's a huge debate. Producing visible light is metabolically expensive, right?
Oh, very. It requires burning energy that could be used to grow.
So in the harsh forest, why expend energy to glow? Especially since the glowing mycelium is hidden deep inside the wood. It's not signaling a mate like a firefly.
Right. Or luring prey like an anglerfish. For a long time, it was a total mystery. But the prevailing theory now is that the light itself isn't the point at all.
It's a byproduct.
A byproduct of antioxidative metabolism. Remember how degrading lignin is violent on a cellular level?
Yeah, it produces massive amounts of dangerous reactive oxygen species, or ROS, primarily peroxides.
Right. And those peroxides are highly volatile. If unchecked, they cause cascading cellular damage, destroying the fungus's own DNA.
So the peroxides are toxic waste generated by its own digestion. The fungus is poisoning its own environment as it eats.
Precisely. So it desperately needs an antioxidant defense mechanism. The oxidation of luciferin by luciferase acts as that detoxifying process. It neutralizes the dangerous ROS.
I love this theory so much. The photon of light is just the physical exhaust fume of neutralizing cellular poison.
It's the glowing exhaust of a fierce microscopic engine.
That's incredible. Now, for millions of years, that exhaust fume was just a quirk of forest ecology. But this brings us to one of the most exciting parts of the deep dive.
The modern materials science.
Yes, because human scientists have figured out how to hijack this caffeic acid cycle to create living technology.
The breakthrough research out of EMPA in Switzerland, they've created what the literature calls a hybrid living material, or an HLM.
Basically, they built a biological light bulb.
That's a good way to conceptualize it. They use a block of balsa wood as the structural filament, and the electricity, the power source, is the living Desarmillaria tabescens fungus pulsing inside.
But creating this glow wood is highly technical. You can't just rub spores on a stick.
No, it's a very exacting recipe to maximize light production without destroying the wood.
So let's walk through that recipe. They specifically chose balsa wood. Why balsa?
Balsa is incredibly low density, but structurally highly porous. It has massive empty cellular cavities. That's vital because the first step is intense water saturation.
Right. They put the blocks in a vacuum pressure chamber and impregnate them with water until they reach a moisture content of 700 to 1200%.
It's thoroughly saturated, way beyond a normal rainstorm. Then they autoclave it to sterilize it.
Okay, so sterile, super wet balsa blocks. What's next?
They submerge them in an aqueous growth medium containing 3% microfibrillated cellulose, or MFC.
MFC acts as easily accessible food, right?
Yeah, a nutritious carbon source, so the fungus can establish itself before it has to eat the solid wood. They introduce the living mycelium into this bath and let it incubate for 3 full months.
3 whole months of just slowly eating cellulose, building the melanin rind, sending down snorkel pores. But while it's submerged for those 90 days, it doesn't glow at peak intensity, does it?
No, it's relatively dim. It's metabolizing but holding back. To turn the bulb on, there's a magic trigger.
Controlled dehydration.
Exactly. They remove the colonized block, brush off the MFC, and expose it to open air in a dark room.
And as the water slowly evaporates over 4 to 10 hours,
Oxygen rushes in through those aeration pores. That sudden influx of oxygen hits the huge reserves of luciferin the fungus stockpiled for 3 months.
and boom.
Boom! The luciferase catalyzes it all at once, emitting a steady, powerful glow. It's so strong the researchers could read lettering carved directly into the glowing wood.
That visual is stunning. But reading this study, something bothered me. We established this fungus is a destructive pathogen that smashes wood into a slurry.
Right.
So after 3 months of being aggressively eaten, why doesn't the balsa block crumble to dust? How is it still solid wood?
That's the stroke of genius here. It comes down to selective delignification.
Explain that selective eating.
Wood is primarily lignin, hemicellulose, and cellulose. Our fungus preferentially targets and degrades the lignin and hemicellulose, the glues, to fuel its caffeic acid cycle.
But it leaves the cellulose largely intact.
Crucially intact. And cellulose is the fundamental structural scaffolding, so the wood retains its physical shape and solidity even while colonized by a light-emitting organism.
So the physical bulb remains intact while the light emanates from the empty spaces within.
Exactly.
This is so much more than a lab trick. The implications are massive. Biodegradable, zero-energy ambient lighting.
Imagine soft interior lighting built directly into wooden architecture, or eco-design products that don't need lithium-ion batteries or a fragile power grid.
It could cut domestic electricity consumption, lower CO2 emissions, and reduce light pollution in cities, since the glow is soft and non-invasive.
And at the end of its life cycle, you don't throw it in an e-waste landfill. You toss it in the garden, and it biodegrades perfectly.
It completely flips how we think about manufacturing. The material itself is a living agent. But scientists in Swiss labs aren't the only ones exploiting this biological machinery.
Not at all. Deep in the ancient forests, organisms have been hacking this exact fungus for millions of years.
Which brings us to the hidden world of fully myco-heterotrophic plants.
A story of highly specialized, ruthless symbiosis. We're looking at the relationship between our fungus and certain rare orchids, like Cyrtosia septentrionalis.
These are the ghost orchids, right? They've completely given up on photosynthesis. Zero chlorophyll.
Yep, they are obligate botanical parasites on fungi. They steal all their carbon from the soil, and the germination requirements for their microscopic seeds are notoriously difficult.
The sources mention they need atmospheric terraforming.
It's an apt comparison, like building a colony on Mars. The seeds won't germinate in normal 21% oxygen air. They need a highly localized microaerophilic environment.
Oxygen reduced to between 13.1 and 17.0%, and carbon dioxide elevated to between 2.7 and 4.9%. Those numbers are incredibly specific.
Very specific. And out in the wild, who creates that bizarre atmospheric pocket?
Desarmillaria tabescens.
Purely as a byproduct of its intense wood rotting, it consumes massive amounts of oxygen through its snorkels and exhales dense plumes of CO2, terraforming the dirt around it.
Creating the exact trigger the orchid sees needs to wake up. But it gets even crazier with this phenomenon called the mycobiont shift.
Oh, the manipulation is ruthless. When the seed first germinates, it doesn't partner with our fungus right away. It chemically recruits a fast-acting rotter like Physosporinus for an immediate sugar rush to establish its roots.
But Physosporinus burns through the local nutrients too fast.
So the orchid has to execute a mycobiont shift. It essentially fires its first fungal partner and recruits a secondary colonizer for the long haul.
Desarmillaria. But how does a fragile plant convince a destructive pathogen to let its nutrients be stolen without just getting eaten?
A delicate chemical dance. The orchid secretes fungistatic compounds that regulate the fungus. It invites it into its root tissue, but blocks it from the vascular core.
It builds a corral inside its own body.
Traps the fungus inside, and then taps into its slow-burning nutrient network, stealing carbon to sustain itself for years.
Talk about a plot twist. This feared tree killer gets perfectly manipulated by a ghost orchid into a beast of burden.
It illustrates the staggering complexity of forest ecology.
So we've seen it terraform soil, rot wood underwater, glow in the dark, get parasitized by orchids. How is a single organism capable of all this?
It all comes down to the hidden chemical factory coded into its DNA. The genomic mining reveals a massive terpenome.
It produces a complex class of compounds called meliolides, right?
Protooludin aryl esters, yeah. And they have potent anti-tumor, antibiotic, and cytotoxic properties. In labs, they literally stop human cancer cell lines from growing.
And the enzymatic machinery that builds these is fascinating. The genome sequencing uncovered 12 putative sesquiterpene syntheses, or STSs.
And 6 of them belong to a completely novel evolutionary group called clades the 4.
The literature calls them highly promiscuous producers. What exactly does that mean in biochemistry?
Think of a standard enzyme like a machine that only folds one type of paper airplane. Rigid precision. A promiscuous enzyme is more like a master origami artist.
Oh, okay.
It takes the exact same base precursor molecule, and depending on tiny shifts in the local microenvironment, like acidity, it folds it differently. One fold makes a swan, another makes a frog.
That is an incredibly efficient way to build a chemical arsenal. One flexible enzyme making 50 different complex structures depending on what the fungus needs.
Exactly. For example, the DTSTS10 gene primarily produces alpha-hemocyaline, plus several structural variants. And the beauty is, we don't have to harvest rotting logs to get these chemicals anymore.
Because we have the genetic blueprint, we can outsource the manufacturing.
Scientists are taking these promiscuous genes and inserting them into standard laboratory yeast, Saccharomyces cerevisiae.
The exact same yeast used to make bread and beer.
Yeah, turning brewers' yeast into a microscopic factory pumping out rare anti-tumor fungal chemicals.
We are hijacking fungal chemistry to brew tomorrow's medicine. That is staggering.
It's an incredible time for this field.
So let's take a breath and recap the sheer scope of what we've explored. We started 51 million years ago tracking a ringless ancestor.
Surviving the PTM.
Right. Then it masters structural destruction with a melanin snorkel to breathe underwater. We explored how its glowing exhaust fume is being used to build biological light bulbs out of balsa wood.
And the ghost orchids manipulating it.
Yeah. And finally, its promiscuous enzymes brewing cancer treatments in yeast. It's a profoundly dense resume for an organism most people haven't heard of.
And I want to leave you with one final thought on where this is heading. There's a quietly revolutionary concept hidden deep in the sources.
Oh,
We know our fungus has a massive mitochondrial genome, and separately, scientists have developed an ATMT system specifically calibrated for these fungi.
ATMT, what does that practically mean for us?
It means we can edit their genes. It's an agrobacterium tumefaciens-mediated transformation system. They use a bacterium as a microscopic delivery vehicle to permanently alter the fungus's genome.
Wait, really?
Yeah, they can silence target genes or insert foreign ones, like making it glow fluorescent green under UV light. We possess the tools to cleanly engineer this organism.
Wow. Okay, if we combine all of this,
Follow the logical trajectory. If we combine genetic engineering with its ability to structurally preserve wood while glowing, and its massive chemical factory that synthesizes anti-tumor compounds,
are we seriously looking at a future where we can print a living biological house?
it's no longer science fiction. It's an engineering hurdle. Imagine growing structural beams that maintain the strength of solid wood, but ambiently light your living room with zero electricity.
And walls that actively purify the air through those microscopic melanin pores?
And because we can edit the genome, maybe those walls gently off-gas therapeutic anti-tumor compounds or bronchodilators while you sleep.
A house that lights itself, breathes for you, and chemically heals you, all powered by the ancient machinery of a ringless mushroom that survived the thermal maximum of the Eocene epoch.
It's definitely a thought to linger on.
It really is. Thank you so much for joining us on this deep dive. The next time you're walking through the dark woods and you see a dead, rotting stump, take a much closer look. You might just be looking at the foundation of our future.